Hello everyone, good evening. So I hope I'm audible to all of you. So today we will be talking about or discussing some questions from your unit from unit 3, right? And we will be discussing some 10 questions from here. So I hope you're all ready. So mainly we'll talk from your replication part, all right? And we will also talk about this DNA repair. Okay, so the first question that we have here is a plasmid with linking number of 200, topological winding number of 200, and a writhing number of 0 was transformed into E. coli. The plasmid was re-isolated from the culture of the transformant. The re-isolated plasmid was found to possess a writhing number of -5. Following statements are made about this observation. So these are your CSIR questions, and this is one of my favorite questions, to be very honest. The question is very simple, and it is being stated from the concept of topology, DNA topology. And this is a very simple question if you know the concept. So you have to have the concept of DNA topology. What is this? One second. Students, am I audible? Okay, right. Sorry, I was not getting that reply of yours. So I was okay. Now I can see your replies. Right. So anyone would like to try and give me the correct answer here? Let this be a question answer discussion kind of thing, right? So I can give you a hint that this is going to be the formula for this is linking number is equals to twist plus write. And this is very important. This concept of twist and write is very important because it is around this concept that your topoisomerase enzyme is working during your replication process. The entire work of the topoisomerase is revolving around this concept of linking number, twist, and right. So what is this twist? It is the number of times one DNA strand is going around the other DNA strand, or you can say it is crossing over the other strand. That is a twist. And when the entire DNA, it wraps around itself, that is the right? So in this case, the correct answer will be option 2. Why is it option 2? Because I told you it is going to be linking number is given here as 200. Twist number is given here as 200, right? Initially, there was no right, but now there is a right involved, which is of -5. So if you have a right included here, automatically 200 minus 5 will give you 195 as your linking number, right? So therefore, statement 1 is correct. The linking number of the reisolated plasmid will be 195. If 1 is correct, obviously 2 is wrong. Okay. Coming to the third statement, twist of the reisolated plasmid would remain 200, and that is what we are keeping, all right? Twist is 200, a right has been introduced, so that will have an impact on the linking number, not the twist. So C is correct, and twist of the reisolated plasmid is 195. This is wrong. So if C is correct, D is wrong. All right. So you get a lot of questions from this concept of linking number, twist, and right. Next, this is a question from your tRNA. And as you know, we have the polymerase, the RNA polymerase, which transcribes your tRNA as well, right? Now, can someone tell me which is the RNA polymerase that transcribes tRNA? So here comes the entire concept of the different RNA polymerases, what they do, what kind of promoters they have, and how they synthesize these different RNAs, the mRNA, the tRNA, the ribosomal RNA, what happens to them. This question centered around all those ideas. So it is saying that the following statements are made with reference to the fact that tRNAs are known to possess T in their sequence. T means thymine, right? Now, why have they pointed this thymine? Because obviously, as we know that RNA, they don't have thymine. They have uracil at the place of thymine, right? But this tRNA has thymine. So what can be the possible reason for having that? So the options given here are RNA polymerase 3 utilizes TTP as one of the substrates. Like any other RNA polymerase 3 also utilizes RUTP, but when the RNA polymerase 3 reaches a looped structure, it binds to S-adenosyl methionine to methylate C5 position of the incorporated uracil to result in a thymine in a co-transcriptional manner. Number C, during transcription of tRNA genes at the designated positions, DNA polymerase replaces RNA polymerase 3 to incorporate T in the tRNA transcript. A specific methyl transferase utilizes a methyl group donor to post-transcriptionally modify the specific uracil residues into thymine residues. Uracil to thymine conversion occurs in a large number of tRNA in the T-Si C loop. Can you imagine the tRNA structure? If you can imagine the tRNA structure, you will be able to relate to this question. Can I get some answers from you? Yeah, I'm waiting for your answers. Give it a try. Anyone? Okay. The answer to this question is option 4. The correct statement here is D and E. So basically, what happens is this thymine, which is 5-methyl uracil, this is normally found in your DNA. Okay, and it is also found in this T sil loop of your tRNA, where it is attached to the ribose and it is made all by methylation of the uracil. Okay, after transcription, the uracil which is found in the T sil loop, that uracil gets modified, gets methylated to give you the thymine here. Okay, so in addition to uracil, even guanine, adenine, cytosine, that can also get modified by methylation. It can happen. But here, thymine is not incorporated during transcription, but it is post-transcriptionally modified uracil. Okay, next going over to the second question. The third question, rather. So it says that in all organisms, it is critical that replication initiation be tightly controlled to ensure that chromosome number and cell number remain appropriately balanced. Given below are several statements regarding the regulation of replication in E. coli. So the statements are given to you. Number 1, hemimethylation and sequestration of ORIC by a protein called SEC-A prevents initiation of replication. Option B, availability of DNA A protein is an important requirement for initiation of replication. Third statement, the ratio of ADP to ATP is important as a high level of ADP is required for initiation of replication. And number D, recruitment of HDA protein by sliding clamp inhibits ATP hydrolysis required for initiation of replication. Which of the above statements are not true? Mind it, they're asking not true. So, what are they talking about? Could you recognize the place from where the question has been asked? Can you recollect the regulation that happens here and why that regulation happens? Because all these things are very, very important. CSIR asks question from each and every details, each and every regulation. So when it is happening, where it is happening, why it is happening, these are very, very important things to be considered. So who's going to give me the correct answer here? Okay, the correct answer here is answer 3. Okay, a statement the option 3. So let's see what they are saying. Because see, they are asking not true. Okay, so you have to be very careful about this. Many people go wrong in understanding what they are asking, true or not true. So first statement, they are saying hemimethylation and sequestration of ORIC by a protein called SEC-A prevents initiation of replication. This is absolutely true. We need to regulate replication only once in a cell cycle, right? We need to restrict that. In order to restrict that, we take the help of SEC-A, because for the initiation of replication, you need to recognize this ORIC, and that is done by DNA-A protein. But if SEC-A is bound to this ORIC, then obviously DNA-A protein cannot bind to it. Okay, and if DNA-A protein is not binding to ORIC, it will not initiate replication. So statement A is correct. Statement B, availability of DNA-A protein is an important requirement for initiation of replication. Just as I explained, DNA-A protein is important. The ratio of ADP to ATP is important, as a high level of ADP is required for initiation of replication. No, this is wrong. Initiation of replication requires ATP, okay, not ADP. Recruitment of H-duct protein by sliding clamp inhibits ATP hydrolysis required for initiation of replication. Again, this is wrong because ATP hydrolysis is required. Okay, so therefore the incorrect statements are C and D. So my correct option is option 3. Now moving forward to question number 4. I hope you are trying, all of you, because until unless you try, you will never know how much you have prepared and how much more you need to prepare. So the exam is just round the corner. It is right in Feb. Okay, it's almost mid of Jan. So until unless you try, the entire discussion is futile, basically. So this is something, not just a discussion. This is also, you can say, it is your own test of knowledge. Okay, coming to question number 4. So the total RNA was isolated separately from cytosol and nuclei of human cells growing in a cell culture. Be very careful about the statements. Total RNA was isolated separately from cytosol and nuclei. Okay. Each sample was mixed with a purified denatured fragment of a DNA corresponding to a large intron of a housekeeping gene and incubated under renaturing condition. Given below are the statements made about the outcome of the experiment. So statement 1. RNA isolated from nuclei will form RNA DNA duplexes because of the presence of introns in the primary RNA. Statement B. Cytosolic RNAs usually will not form RNA DNA duplex. Both cytosolic and nuclear RNA will not form RNA DNA duplexes as transcription and splicing occur simultaneously. Option D. Cytosolic RNA will form RNA DNA duplexes because unspliced cytosolic RNA are exceptionally stable. Which of the above statement is or are more likely to be true? So I hope you have understood what they have done. They have taken out the total RNA from the cytosol and from the nuclei. Okay, so you have two sets of RNA. With both the sets, you are incubating a DNA sequence which is corresponding to an intron. Okay, an intron of an housekeeping gene. So that means it is like universal. It is definitely, the sequence is definitely going to be present in that particular cell. Okay, or in that particular genome. So When you are doing that, what kind of outcome will you get to see? Will the RNA, which is already a single strand, and the DNA of the intron, you have already denatured it, so now it has become single strand. So, will the single stranded DNA bind with your single stranded RNA or not? How will it bind? That is a question. Only when it gets a complementary sequence, right? Because ATGC has to match. So when it gets a complementary sequence, only then this denatured fragmented DNA will be able to bind to the RNA. Now the question is, this DNA has been taken from an intron part, okay? And you have taken out the total RNA from cytosol and nuclei. So is there any difference between the cytosolic RNA and the nuclear RNA? That is the main concept that you need to understand here. And for that, you need to study this post-transcriptional modifications, all the things which comes under post-transcriptional modifications. So that will involve your splicing, your capping, polyadenylation, right? Though over here, splicing is what is the main idea. So again, anyone wishes to give me the answer? See, these questions, Parsi questions, they are all concept based questions. You can't get away by just memorizing the facts. You need to analyze, you need to visualize what is happening. You have to understand each and every bit of it. Okay. So the correct answer for this is option 3. Which of the above statements are or is are most likely to be true? First and foremost, the statement RNA isolated from nuclei will form RNA DNA duplexes because of the presence of introns in the primary RNA. Yes, when the RNA is being formed, the introns are present, which one by one will be spliced out as your transcription proceeds, right? But there is a high chance that this this RNA has been isolated right before this particular intron could get spliced out. All right. So there is a chance that this RNA and DNA can form a duplex. Second statement, cytosolic RNA usually will not form RNA and DNA duplexes. Why? Because by the time RNAs have moved to the cytosol, or it has been exported from the cytosol, all the introns has been spliced out, because otherwise it is not possible for the RNAs to come to the cytosol. The capping has to be there, the polyadenylation has to be there, and some studies do mention that splicing needs to be completed before RNA can get exported to the cytosol. Okay, according to some the studies. So that is why A and B is correct. C and D is not correct. Okay. Now, this is again a very simple question, part C question, but this is something which is talking about the function of the enzyme. So there are lots and lots of enzymes in your unit 3, and you have to memorize the function of each and every enzyme as well as the minimum structure. Okay, you don't have to get into the details of the structure, but the minimum structure is what is needed to be memorized. So here they have given some sets of enzyme in column A, and they have given some sets of enzyme in column B. All right, so you have DNA topoisomerase 1, DNA topoisomerase 2, polymerase epsilon, polymerase delta. And under column B, they have given synthesis of Okazaki fragments, leading strand, synthesis, double strand break and ligation, single strand nicking, all of these. So what should be the answer here? Come on, this is something which I can expect from all of you, isn't it? This is not a very difficult question. Though it is a part C question, it is less of analysis, more of memorization. So who all is going to give me the answer here? No? Okay. The correct option here is option 1. So basically topoisomerase 1, this is required for single stranded break, or this can only make single stranded breaks. Topoisomerase 2 can do double strand break. Polymerase epsilon is for your leading strand synthesis, whereas polymerase delta is for the lagging strand synthesis, that is the synthesis of the Okazaki fragments. Okay? Okay. Moving on to another question, question number 6. So, following statements were made about the post transcriptional processing of RNA in eukaryotes. First statement: soon after transcription initiation, RNA polymerase 2 pauses 30 nucleotides downstream from the site of initiation until the cap structure is added to the 5' end of the nascent pre-mRNA. Statement B: the 5' splice sites are functionally divergent, whereas the 3' splice sites are functionally equivalent. Option number C: in addition to helping in recognition of the splice size, the exon definition also functions as a splicing regulator by allowing pairing and linking of adjacent 3' splice site and 5' splice sites. The intron definition mechanism applies only to the larger introns and assists in achieving alternate splicing. The splicing reactions carried out in vitro have revealed that the first and the second transient esterification reactions are reversible. So, which of the following combination is correct? So, again, this is from your post transcriptional modifications, right? So, you have to have a good idea about all these splicings and all. So, what do you think should be the answer for this? So what is an exon and what is an intron? So who's going to tell me this? Okay. The correct answer for this is option 4. Okay. Soon after transcription initiation, RNA polymerase 2 pauses 13 nucleotides downstream from the site of initiation until the cap structure is added to the 5' end of the nascent pre-mRNA. That is true. In fact, during the initiation, you have the transcription factors or the initiation factors which initiates the transcription, but later on for the forward movement of the polymerase, you need an elongation factor as well. Until and unless that elongation factor comes and modifies or phosphorylase the second serine of your this polymerase tail, the elongation process will not happen. In the meantime, what is happening? The cap structure is getting added, right? So statement A is correct. Statement B is saying that the 5' splice size are functionally divergent, whereas 3' splice size are functionally equivalent. No, these are not. Statement C is saying, in addition to helping in recognition of the splice size, the exon definition also functions as a splicing regulator by allowing pairing and linking of the adjacent 5' and 3' splice size, which is true. And mostly these exon definitions and all, no, they come in very handy when it is when they are doing the alternative processing, okay, or alternative splicing. During that alternative splicing, these intron definition, exon definitions, these come very handy. They actually help in the precision. So A is correct, C is correct. Now D is saying intron definition mechanism applies only to the larger introns. No, this is not right. And then we have the splicing reactions carried out in vitro have revealed that the first and second esterification reactions are reversible. That is absolutely true. The esterifications reactions are uh, reversible reactions. Okay. Now moving on to question number 7. So question number 7 is saying that in an in vitro experiment using radio-labeled nucleotides, a researcher is trying to analyze the possible products or DNA replication by resolving the products using urea polyacrylamide gel electrophoresis. In one experimental setup, RNase H was added, while in another set, no RNase H was added. The possible observation of this experiment could be what? So you have to exactly know what is a function of RNase H in DNA replication, whether it is functioning in prokaryotes or eukaryotes, like wherever it is functioning, what it is doing, how it is doing. Okay, only then you will be able to answer this question. So the first statement says that there is no difference in the mobility of labeled DNA fragments between the set 1 and set 2. So set 1 is where the RNase H has been added. In set 2, RNase H is not present. Okay. There is distinct difference in the mobility of the newly synthesized labeled DNA fragments between set 1 and set 2. Next, the mobility of the newly synthesized labeled DNA fragments in case of set 1 is faster as compared to set 2. The mobility of the newly synthesized labeled DNA fragment in case of set 1 is slower as compared to set 2. So again you have to say which represents the correct observation. So recollect what is the function of RNase H. What was this RNase H doing? Anyone would like to give me the answer? Okay. The correct answer is going to be, should I tell you the answer or you're still thinking? The correct answer here is option 2. See, first, what is the function of RNase H? RNase H is helping in the removal of the it is helping in the removal of these primers, right? And where will you get to see more primers? Obviously, it is in your uh, the lagging strands. Okay. Now, these primers, they are the RNA. It is an RNA strand. The primer is actually an RNA strand. Now, obviously, there will be a difference in the mobility of an RNA strand and a DNA strand. Now, wherever RNase H is present, that means these primers, these RNA strands has been removed and replaced by DNA, DNTPs. Okay. But where RNase H is not added, that means the primer is still remaining. That means the RNA section is still remaining. So here lies the difference. So therefore, statement A is incorrect, statement B is correct. There is a distinct difference in the mobility of the newly synthesized labeled DNA fragments between set 1 and set 2. Okay? And what is the difference? The mobility of the newly synthesized labeled DNA fragment in case of set 1 is faster as compared to set 2 because in set 1, the entire strand is made up of the DNTPs. There is no primer present. Okay, everything has been replaced because you have added RNase H. So these mechanisms, you need to understand. You need to know exactly how RNase H is working, the characteristics of these RNase H, FEN1, and there are many more enzymes. Okay? So when you're studying about replication or transcription, just the process is not enough. There are many small details which needs to be clarified and understood. Okay, coming to question number 8. So they have given you a picture here. This is the picture of a replication bubble where you can clearly see that this is a leading strand and these are the lagging strands, right? So in the figure above, replication of DNA beginning from the origin of replication of chromosome of a newly identified bacterium having a double-stranded circular DNA genome is shown. Characterization of a DNA polymerase responsible for genome replication showed that DNA synthesis occurred in 5' to 3' direction, and it depends on the presence of a primer. Polarity of DNA 5' or 3' are as shown. Replication begins at a point marked O, which is the origin obviously, on the left of the bubble, and both the parent strands are replicated concurrently. That means simultaneously it is getting replicated. The longer arrow inside the bubble shows the leading strand, whereas the shorter arrows show the Okazaki fragments. So, based on this, they have given you four statements, or these are the options you can say. So, you have to select the correct statement out of the four. Observe the picture very carefully, understand, relate the picture to each and every option. Mainly, they are talking about two things over here. They are talking about the mode of replication, whether it is bidirectional or unidirectional mode. And they are asking about the Okazaki fragments, which is the first Okazaki, which is the second, which is the third. These are the two ideas or the two concepts that they are asking here. So, again, analyze this and tell me what should be the answer. Okay. First and foremost, look at where this origin is present. It is not at the center, it is marked at one side, right? So that means it is moving towards. So here there is only one leading strand shown, right? And one lagging strand is shown. And it is like moving towards only one direction. It's not moving at two different directions. Moreover, the origin is here, and you can see this is the direction of your leading strand. So this clearly states that this is your uh, unidirectional mode of replication. Okay. Now, if this is the leading strand, that means your helicase should be present here. Okay, so the helicase also has moved from this origin side to this position at the present. So therefore, which is the first Okazaki fragment that has been synthesized? It is this one, right? So the correct answer here is option 3. It is a unidirectional mode of replication wherein synthesis of the Okazaki fragments marked C occurs prior to those marked A and B. Okay? Now going over to the ninth question. So during maturation process of some RNA molecules, formation of 2' 5' phosphodiester bond takes place. Following statements are made about this phenomena. 2' 5' phosphodiester bond. When do you get these phosphodiester bond? 2 to 3. Normally, what is the polarity of the phosphodiester bond? In a DNA, what is the polarity of the phosphodiester bond? Where is it present? Where do you get 2 to 5? And in relation to this, I can ask you another question. Where do you get 5' to 5' phosphodiester bond? All of these are very important. The bondings, the polarities. So again, the statements they have given you here is spliceosome mediated removal of intronic sequences occurs through the formation of 2' 5' phosphodiester bond. Removal of group 2 introns occurs through the formation of 2' 5' phosphodiester bond. Enzymatic removal of introns from the yeast tRNA precursors involve 2' 5' phosphodiester bond. RNase P mediated 5' and maturation of tRNA precursors involves formation of 2' 5' phosphodiester bond. So, which is the correct statement. What do you think? So normally the phosphodiester bond is your 3 prime 5 prime, right? In your DNA it is 3 prime to 5 prime, that is a phosphodiester bond. In the case of your RNA capping, you will get to see the 5 prime phosphodiester bond. In splicing, you do get to see this 2 prime 5 prime, but in which splicing, which kind of splicing? There are four categories of introns, and accordingly, you have four different methods of splicing. Which method will you get to see this 2 to 5? Again, I'm telling you, for every mechanism, for everything, so you have to go into the details because you never know from where the CSI is going to ask questions. Okay? And the pattern is changing, the concepts they are twisting. So you have to have a very thorough knowledge about each and every process. The correct answer here is option 3. Spliceosome mediated removal of intronic sequence occurs through the formation of 2 to 5 phosphodiester bond. Do you remember there was this internal adenine, the 2 prime site, right? So the spliceosome was one was binding at the 5 prime site, the second one was binding at this 2 prime branch point. Okay, so the spliceosome mediated intron, they will show 2 to 5. Group 2 introns will also show 2 to 5 phosphodiester bond. Okay, and nowhere else will you get to see this 2 to 5. In both the cases, you have this internal adenine which act as your branch point. All right, and coming to the last question. The last question is saying the complex responses to different of DNA damage in both prokaryotes and eukaryotes fall into three main categories: damage bypass, damage reversal, damage removal. Many repair proteins are isolated, like DNA methyltransferase, DNA glycosylase, RNA polymerase 4. So, which of the following statements the correct combination is given? So, this is all talking about DNA damage. So you have the base excision repairs, you have the nucleotide excision repair, you have the direct repairs, you have the double strand break repair, single strand break repair, translation is there, right? There are so many processes, there are so many enzymes, there are like different processes happens in different cases. So I'm expecting some answers. This is the last question. At least give it a try. Come on. If you keep quiet, then I would presume that you don't know the answer. And if that is so, then why don't you know the answer? You haven't prepared? This is new to you, these parts? You need to go through, or you haven't gone into such depths? So these are questions which I'm not asking actually. This is something that you should ask yourself. Okay, because CSIR, the level is the standard of the questions, it is becoming tougher with days. Okay, and if you don't have a very clear concept, very clear knowledge as to where to study, what to study, how to study, then it becomes very difficult to manage all the 13 units and prepare for your CSR exam. It becomes very difficult. So, this is a very basic thing. Okay, Leo has given an answer here. So the correct answer is option 3. Very good. So out of so many students, I find Leo giving me this answer. So basically, you have damage bypass is done by your DNA polymerase 4. Damage reversal, this is a direct damage reversal, also you can call it. It is done by DNA methyl transferase. Damage removal, it is by, there are many enzymes. One of it is your DNA glycosylase, which you use it for your base excision repair. Okay. So that was my set of questions for discussion. I hope you have understood where to study, how to prepare, right? If you still need any guidance, do contact us. We do have this Lakshya batch, and enroll with us. We will definitely help you in your preparations. So that is all from my end today. Thank you and good night.